All right, so we've got how bacteria can survive. We've got these structures that will protect them. We've got how they can move around. Now it all comes down to the name of the game, which is reproduction and survival. If you can reproduce and survive, you win life. That is, people are always like, what's the meaning of life? Reproduction and survival. There it is, it's solved. No more problems there. Bacteria can reproduce and survive in several different ways. And I want to go over a few examples. When we're talking about reproduction, binary fission is how it works in bacteria. They split. This is the process of splitting into two identical cells. Now I'm going to split hairs. I'm splitting, right? Split hairs here. People often say this is mitosis, but it can't be mitosis. Why is binary fission not mitosis in prokaryotes? Exactly. Mitosis is the division of the nucleus. The division of the cell is cytokinesis, or in this case, binary fission. I mean, that's, I don't want to have to go into all of the parts of, first it reproduces the DNA and so on. You guys took Bio 101. You know, basically, you reproduce the DNA, you grow, you reproduce the DNA, you check out the DNA, you split the cell in half. In order to survive in times when resources are short, you might build an kinetase. These are large food-filled cells. Literally, you'll have a group of cells. In this case, it's a streptobacillus cells. You'll notice there's a long chain of these rod-shaped cells. One of them grows to enormous proportions because during times of plenty, the bacteria keep pumping food into that cell. It's like a storage bin. One big storage area for the cells. When times get harsh, the guys on the outside start to die off, but the stored one survives. The one that stored all that food survives. It is able to even push food out to the other cells. What's really interesting to me about this is, imagine then this chain dying off really low because it's running out of food. Eventually, when the food comes back or times get a little bit better, that one cell is able to start to undergo binary fission and reproduce the whole group. So kinetase gets cells through times of hardship. But that's just with the nutrients aren't available in the environment. Sometimes the environment itself gets bad. Sometimes the bacteria cannot survive in the environment that it's found itself in. In that situation, the bacteria might surround its nuclear material with what's known as an endospore, sort of a structure within the cell to protect the most vital components of it. When the rest of the cell dies off, the endospore survives in a state of stasis. It doesn't do anything until that endospore encounters positive conditions again, at which point it opens up and the cell begins to reproduce rapidly. The best way I can explain this to you is you guys have been sick. You've been coughing. Heck, you may have been coughing lately. There's something going around. I know, right? When you're sick, coughing things up, you may cough into your pillow because you're sick and you're tired and we don't want to do things. So you're like, and you get this phlegm with bacteria on your stuff. After you're done being sick, you wash your beddings. Or not. I don't know what you guys do. A lot of people tend to wash their pillowcases and their blankets and things. One reason you do that is because when you are coughing, you hacked up bacteria that can no longer survive in your lungs because there's not a moist, happy place for them. So it lands on your pillow. That makes sense? If you don't wash that, you put your nose on your pillow and you breathe in when you're sleeping. What happens, all of those endospores that had formed on the pillow get sucked into your lungs again, where it's a nice, happy environment for them. They open up and they begin to rapidly multiply. So endospores provide an ability for a bacteria to survive in harsh conditions for a long period of time and then suddenly come back awake. This happened in the Arctic. We are not the smartest species. We found bacteria from the Cenozoic, Paleozoic. We found really old bacteria that had survived as endospores. And so our brilliant mode of thinking was, hey, let's culture these. Let's give them the perfect environment to grow in and see what happens. And they did. And now we've got some bacteria that we thought went extinct millions of years ago, sitting in a lab. Don't know what they'll do. Don't know how they'll react with things. But we got them. Because of these endospores, they survived that long. No, you just run tests on them. They haven't escaped the lab or anything. That's like a sci-fi movie waiting to happen. But bacteria have got to get food somehow. They've got to eat. They've got to take in nutrients. And there are 4 methods. You guys had this question on your lab. You might have the autotrophs, whether chemoautotrophic or photoautotrophic. Chemoautotrophs, they're going to take in inorganic carbon, and using that source of energy, they're going to fix it into organic carbon. So they're not using light, they're using electrons they steal from other molecules, and they build organic molecules from that. This happens down at the bottom of the sea. Photoautotrophs, photo meaning light, they take the energy from light and convert it into the bonds of, use it to convert it into the bonds of glucose or something. They take inorganic carbon using lights and turn it into organic carbon. Never mind, that's what these slides say. Yes, I can. I can absolutely say it, in fact, 2 more times. Chemoautotrophs utilize energy from inorganic molecules, the bonds of inorganic molecules, to form organic molecules. Good on that one? All right, next one. Photoautotrophs take the energy from light to form the chemical bonds. They take inorganic molecules and convert them into organic molecules. Next, we have our heterotrophs, our photoheterotrophs, and our chemoheterotrophs. Again, photo means light, chemo, think chemical. And they're heterotrophic, which means they're going to require organic molecules. First group, photoheterotrophs. The photoheterotrophs can use light to produce ATP. They absolutely can. But they have to take in organic molecules in order to build other organic molecules. So instead of taking light and fixing inorganic molecules, they're taking light and fixing and using the organic molecules to make more organic molecules. If it's green, it's probably taking in light. Then you've got your chemoheterotrophs. They use energy from organic molecular bonds to form new organic molecular bonds. You are a chemoheterotroph. You don't get to make your own food. You have to take it in from the environment. So those are the 4 ways that bacteria can get nutrients. Bacteria are also going to interact with oxygen in several different ways. And we've talked about some of these words already, which is cool. There could be obligate, aerobe. What does it mean to be obligate? It's necessary. Aerobe, oxygen, so they need oxygen. Absolutely need oxygen. So we got down here. So aerobic. Without oxygen, they don't grow. So the oxygen content at the top is highest. At the bottom, it's lowest. You've got growth only at the top where there's oxygen. You've got your aerotolerant anaerobes. They can survive a bit in oxygen. They do all right, but they really like anaerobic conditions. So they grow better in anaerobic environments, but they can grow slowly in oxygen environments. Because oxygen is such a stupid strong oxidizer. It steals electrons like crazy. They could be facultative anaerobes. We talked about facultative anaerobe last semester with yeast. It prefers, it's going to prefer oxygen, but where there's not oxygen present, it will switch over and undergo fermentation. So you're going to have growth everywhere, which is what this tube is showing. Facultative anaerobes do really well. What kind of cells in your body are facultative? They don't stop when the oxygen stops. All right. Even better than that. You take a deep breath, right? Is your heart still beating? Yeah, what happens when you run out of oxygen? It stops, but only after a while. Before that, it undergoes fermentation in order to keep going. Your body is like, we need to survive. Heck, your muscles, right? Any muscle cell you've got. You're running as fast as you can. I don't know, back to our chasing down a gazelle example. You're running as fast as you can. You're almost there. Does your body stop once the oxygen's out? No, you keep running. It just starts to burn because you switched over into a fermentative cycle. You're still getting energy. And then, of course, we have our obligate anaerobes. Obligate, of course, means needs, and then anaerobes, no oxygen. Where oxygen is present, they do not grow. So the 4 different oxygen requirements of bacteria. Sure. Obligate aerobes. So they need oxygen. Where oxygen is not present, they cannot grow. Aerotolerant anaerobes are going to grow all right in oxygen, but they really prefer low oxygen conditions. Facultative anaerobes grow just fine, oxygen or no oxygen. And anaerobic, obligate anaerobes are going to die in the presence of oxygen. They cannot grow in oxygen. Any questions? Bacteria are absolutely essential and vital to the ecosystem due to their nitrogen fixation properties. This means fixation is something we're going to talk about an awful lot in the course, especially as we get into ecology. Fixation is where you take gaseous nitrogen, in this case, and convert it into an organic molecule. You put it into a different mode. Nitrogen fixation is going to be important for creating the amino group, the functional group, the NH3. Without it, plants, fungus, we cannot build amino acids, and with no amino acids, we cannot build proteins. The only way we have of fixing nitrogen is with bacteria. So typically, bacteria and plants form a sort of harmonious relationship, a symbiotic relationship, where the plant provides the bacteria with some sugar and maybe some housing, and the plant in turn gets ammonia from the amino groups from the bacteria, which will allow the plant to make proteins and grow. Then we come along, we eat the stuff. So here we got a nice cycle going on. Nitrogen-fixing bacteria produces ammonia. That allows for the plant to take up the ammonia to produce polypeptides. When the plant or the animal then dies, those are released as gases to the atmosphere, which the bacteria can then in turn use to fix more nitrogen. So they produce, they turn N2 into NH3, and that turns into an amino acid. Without bacteria, this could not happen. Without bacteria, we cannot make proteins. which is why oftentimes when you pull up a plant, you're going to see these little nodules in the roots. Those nodules are where the bacteria live. These plants are actually producing, in some cases, anaerobic conditions just so the bacteria can survive. Some bacteria, if they don't have the relationship with plants like that, where the plant produces the environment, the bacteria will produce a cell called a heterocyst. This heterocyst cell actually provides it's like a little chamber in which oxygen is pushed out so the bacteria can survive.